Hello, everyone, and Happy New Year. Welcome to Schmidt Ocean Institute's first Back Ashore of the year as we kick off 2025 to learn about life below the seafloor. My name is Allison Miller. I'm a senior manager of Schmidt Ocean Institute's research portfolio, and I would like to welcome everyone listening in to this Back Ashore. If you haven't joined a Back Ashore before, this is a platform and a program where we revisit expeditions that took place on board Schmidt Ocean Institute's research vessels Falkor and Falkor 2 to learn about the scientific outcomes, discoveries, and new knowledge gained by the science teams once they have returned to shore and had a chance to analyze the data and publish their works. Today, we are revisiting an expedition from 2023, the Underworld of Hydrothermal Vents, which discovered animal life in the shallow crust of deep sea hydrothermal vents. And the team just published their results in Nature Communications in October. So we're really excited to hear about their outcomes and what they have been up to. We are joined by 4 of the team members from that expedition, and all were co-authors on that paper. Monika Bright from the University of Vienna, who is chief scientist of the expedition and lead author on the paper. Sabine Gollner from the Royal Netherlands Institute for Sea Research and an expert in deep sea hydrothermal vents and faunal biodiversity. Ian Hughes from Harvard University, whose research interests focus on the physiology and evolution of animal microbe symbiosis. And Tinkara Tinta from the Marine Biology Station Piran at the National Institute of Biology, who is a marine microbial ecology. Thank you all for joining me, and Happy New Year. I'm really excited for this discussion. Before we get started, I'd like to remind the audience to put any questions in the chat, and we will make sure that our scientists answer your questions as well as mine. So let's get started and go back to the very start of the expedition. Monika, could you tell us a little bit about the hypothesis you had for life beneath the vents and what led you to submit a proposal to Schmidt Ocean Institute to conduct this work on board Falkor 2?
Hi, everybody. Hi from Vienna in Austria. I have been working on this giant tube worm from the deep sea hydrothermal vents for a very long time. And I had a special interest also in how they reproduce. So you have males and females. And basically, the female releases then tiny little offspring that develop into larvae, and they go into the water. And what puzzled me for a very, very long time was that when the tube worms larvae, they settle, they kind of almost have to swim against the vent flow that comes from beneath out of the Earth's crust. And that was very puzzling me because they cannot swim against this flow. And in addition to that, I worked on those larvae once they settled on the basalt. And therefore, I collected a lot of those rocks in many, many years. And sometimes I found basically that those larvae settled in cracks beneath the surface, actually, and then they grew towards the surface. And that was even more puzzling because even there's the vent flow and they cannot swim against it. And therefore, we had this idea that the larvae might come with the water, the seawater that is getting entrained and sucked into the Earth's crust in this hydrothermal circulation system, and then it mixes with the vent fluid that comes from beneath, and then they get kicked out again at the surface, and there they settle. So this was basically the original idea, but in order to write a big grant proposal and have 20 people on board, I looked for several collaboration partners, and they're all experts in different fields of organisms and on hydrothermal vents also. And we enlarged the idea because it was known that microbes live beneath the surface, so in the subsurface beneath hydrothermal vents. but there were no animals known. And so we had the idea maybe small animals can live there also. And maybe the larvae, they come through it, come through this subsurface and then they settle at the vent. So this was our original idea, which we posted in the end and we tried to confirm.
Yes, well, I'm glad you submitted a proposal to Schmidt Ocean Institute, and I'm excited to have this discussion to learn more about it. Sabine, why is larval dispersal and the ecology and life around these vents important to study, and why was the location that you sailed on Falcor to instrumental in this study?
Yeah, thanks a lot, Alison. So as you could already see on the pictures, and as Monika already explained, like one of the most prominent animals there is the tube-worm Riftia, the giant tube-worm, and it's sessile. Another thing that is important to note is that these hydrothermal vents, they are patchy distributed along these mid-ocean ridges. So if you want to sustain basically a healthy population, want to get genetic exchange, you basically need to ensure that you can travel and disperse from one vent to the other vent. And this is, of course, true for especially for sessile animals, but actually for all the animals that live at these active hydrothermal vents. And we also know that these, or most of the animals are also endemic to this, at least the large ones are endemic to these active hydrothermal vents. So that's why this larval dispersal is really essential to maintain a healthy population. And especially also at the East Pacific Rise, 90° North, where we studied, we did our studies, it's also an area where we have very frequent volcanic eruptions. So every 15 years, basically, the area is like, basically pathed with lava. Communities are destroyed. Then afterwards, you get new venting again. But then the animals basically need to recolonize again these places. And this is again where we need basically larval dispersal. So, and that was also probably the reason why we selected this place. It's very interesting because of the tube bombs, because of the dynamics there. And it's also interesting, and we choose also this site because we know it very well. So in order to be able to carry out sophisticated experiments, it's good to know at least something beforehand.
Excellent. Ian, once you got to the vessel, how did you set up the ship for all of these undersea experiments? And how did you plan what would happen each day so that you and the team could achieve your goals out at sea?
Yeah, so one of the best things about this cruise, beyond of course the obvious science, which was great, was the organization was really great. So huge shout out to Monika and Sabine for that. And we actually really started planning about 6 months beforehand. And so we all sort of got together on Zooms and we had this big spreadsheet and we actually planned out sort of each specific dive day on the spreadsheet. And we populated it with sort of the areas that we wanted to go because we sort of knew some of the areas on 9.5 North that we were interested in running these experiments. We kind of populated it initially with the kind of canonical experimental things that we wanted to do for the deployment of this experiment. And once we had kind of figured out, okay, what days, what dives we want to kind of survey, what dives we want to, deploy the boxes, pick them up, we basically sort of filled in with some other secondary scientific goals. And so we actually really started that process quite a long time before we even set foot on the ship. And we also, of course, had a lot of chemicals and things we had to ship and get to the Falcor before we started. So that really process began a long time ago, which is really great because we were quite prepared once we got there. Of course, you set foot on the ship and there's tons of unpacking and inventorying and all that sort of stuff. And as Sabina was mentioning, of course, the 9.5 North on the EPR, there's these periodic eruptions. You can never be totally sure if the spots you're hoping to go are still going to be there. So we got pretty lucky and we had to do some surveying initially and try to figure that whole thing out. But another real highlight was that every day we'd have the ROV go down, do some research, whatever our goals were. And then as it was coming up and sort of transiting through the midwater up towards the surface and for recovery, we always sat together in one of the rooms in the Falcor, and while we're sort of watching the light show and all the cool midwater animals, we sort of went around the room with all the science party and discussed sort of what happened that day, what we're planning on doing, and what we're planning on doing sort of the next coming days. And so we had a really good, strong kind of network of planning both beforehand and on the ship to really make sure that everything was going smoothly and make sure that when we were encountering some issues or things that were a little bit more tricky, we were really ready to kind of jump back and figure out what to do.
Thanks, Ian. That's a great reminder to everyone listening. We often showcase the 30 days on board the ship for everyone to see and live stream all the dives, but the planning takes place before and after each dive, well before you even step foot on the expedition. And then, as we're going to talk about, many months and even years after you step foot off the ship. So it's a very long time frame just to plan for those 30 or so days that you're on board. Tinkara, once you got to the research site, my understanding that it took a few trials to get the groundbreaking results that the team eventually published. Could you walk us through some of the first experiments and what you learned from them?
Yeah, thank you. The first experiments were actually, designed mostly by Sabine Gollner and Monika Bright, and I was not really involved in those experiments, so I'll try to do the best I can, and then I hope maybe Sabine and Monika can jump in if I say anything wrong. But they designed this complex system, or like a box, that would allow us to understand if life actually comes from underneath the crust. And this box that was actually placed on the rocks with sealed. Then we pumped in special dye, which allowed us to stain the animals inside the box in this area. And then after some time, we removed the dye, and the animals inside the box remained stained. And then the idea was actually to observe in time if the animals that were larvae that would come from underneath the crust that would not be stained, in fact, no, because they would come from underneath the seafloor. That experiment actually worked very nicely, but then once at the end, when we retrieved the box, we tried to also crush the seafloor to get little pieces of rocks and to see for larvae inside. That was a very difficult task, and it didn't really work as we planned, which is very common in the cutting edge science. So then they tried to drill a larger hole to get access to those larvae, which then finally revealed, even better, or confirmed the hypothesis that we had prior to the cruise, that actually there is life underneath the sea crust. So we actually got like a real proof of concept now. And then afterwards followed several of these trials, or we repeated this breaking or drilling into the seafloor to get a larger set of data.
Excellent. I think that's a good lesson in science and life all around, that there are no failures, just lessons learned. And sometimes when you keep trying, then you get the results that you're hoping for. So it ended up being literally groundbreaking research. Monika, Tinkara talked a little bit about how the team finally achieved the goals that led to the main discovery that we're talking about and that you wrote up. But when it first happened, did you really think that this was going to be such a scientifically leading discovery? You ended up getting a lot of press from the outcomes, and then the paper was covered by Science and of course appeared in Nature Communications. So when you first got the results, what were you thinking?
Yeah, as I remember, it was just crazy, because actually it was the idea of one of the ROV pilots then to finally flip the rock, because we drilled in it, we have a chisel, and then we enlarged basically the crack and took some samples already and temperature measurements and everything, but we felt already, also the pilot felt already, that there's something little hollow underneath, because it was breaking through kind of. And then we enlarged this, and we took a very large pry bar, which was 2.5 meters long. The pilots found it on the ship. that was crucial basically for the final thing, that the pilot then said, okay, I cannot crack it more, but maybe I can flip the rock. And that's what he did. And you saw it on those pictures just now. And when he flipped it, basically it's like you cannot see it on this picture very well now because you have to zoom in a little bit, but you will, yeah, right, like for example here. And here you can see, and that we saw immediately, is that a tube worm has been growing from the roof. It's right in the middle. It's surrounding those so-called lava drops. So they develop when there's a volcanic eruption, and the hydrothermal vent fluid, which is very hot, is basically allowing the lava to flow over it, and then while the lava cools, the drops come, and this basically, on this place, for example, we saw the first tube worms. They were dead actually on the first flipping of rocks, but it meant that yes, the larvae came through with this circulation system. They were sucked into the Earth's crust very shallow, and they settled there and they grew there, and at a certain time they died. And we saw it, Sabine and me and everybody who was in the room saw it immediately, and those were larger ones that were alive then. This is actually the giant tube worm, 50 centimeters long. And so we knew that actually the idea that we had that was so crazy in the beginning, maybe they come from below, they do, but it's not that they might go up and then settle the surface. They probably do this also, but they also can settle and grow and live and reproduce below the surface. And that was completely unexpected. And so basically, yes, the hypothesis was confirmed, but it was even much more, because we found that the deep sea hydrothermal vents, what we know from the surface, they are much larger because they're also in the subsurface, and animals are there. And in the meantime, we have first now identified, that is also already in the paper, we have identified several vent species, so animal species, and that was the discovery. So yeah, crazy.
That's great. I love the line, the crazy hypothesis was not so crazy after all. So that's a great line. And I love that last picture Hannah showed of your reaction when the ROV turned over the vent rock. Does anyone else, any of our scientists, want to share kind of what their reaction was when they were watching the monitors in the ROV control room?
Yeah, so when we're basically like in the RV control room, and we had all these difficulties with collecting this rock, and then finally, like yeah, one of the pilots came up with this idea, and everything actually went pretty fast. We said, yes, why not? We try it. And really, like at the split second this happened, like we could actually see like that there's life below, yeah, in the earth crust, in these cavities, and it was like, I think within yeah, probably not within a second, but within 10 seconds, I think we knew what happened. And I think that was really like a moment where we all got, goosebumps, and everyone was happy. And yeah, it also meant that we had to reschedule also the upcoming dives because of this discovery. So it made our life not easier on board, but very exciting. And really, I think it's like really this also this work together also with the ROV pilots and with all the technicians that really also made this success story, because yeah, it really needs like also the insights of different disciplines to come to these achievements, I think.
Definitely, science is always about teamwork, especially out on a research vessel. Ian or Tinkara, do you have anything you'd like to share about your reactions?
Yeah, I guess I was not in the control room. I was in the lab. But we had big screens in the lab that while you were working, you would like always look a little bit what's happening also in the control room. And it was all of a sudden like you could hear excitement, no? So it was like, so everybody started to run and leave the work and start to watch like really, cool. It's unreal, like a cool movie, that keeps you speechless. And then you just realize that you're actually there. It's not the movie. So you're actually, it's reality for you at the time. You're part of this great moment, in science history, I guess.
Definitely. Ian, did you have anything to add?
Yeah, I think like similar to Tinkara, I was in the lab working a bit, but one of the cool things about the Falcor is, of course, there's like screens like in many places on the ship, and so almost no matter where you are, if the RV is down and some things are going on, you can sort of see what's happening and tune in. And so it was like extremely cool to kind of like walk out of the van where I was working and like look on the screen and just like, wait, hold on, that's underneath the rock? Like they just flipped that over? Which was super cool. And it was awesome to see, yeah, just the whole vibe just like shot up and everyone was really extremely stoked, which is really exciting and fun to be a part of.
Awesome. I love that I can still hear the excitement in everyone's voice retelling that moment of discovery. So that's really awesome to hear. Before we pivot a little bit to talking about coming home and writing the paper and analyzing the data, we do have a question from the audience. So anyone feel free to chime in and answer, but we have an audience member that wants to know if you conducted any geophysical and biogeochemical research during the expedition.
Yeah, I can answer this. So originally we wanted to use a tool that in the end did not work, but we collected, of course, we used all the equipment that was on the ROV. So we had the temperature, the water temperature, the oxygen concentration, pH, salinity, and then we took also samples to water samples and measured also hydrogen sulfide, which is a very important chemical at deep sea hydrothermal vents, because especially for animals like the giant tube worm, they live here in symbiosis with bacteria. And those bacteria, they use hydrogen sulfide and oxygen to gain energy. Also they oxidize it and they fix carbon and share the organic carbon they produce. They share then with the host that has no digestive system. So it's a very important chemical. So that all those parameters we measured.
Excellent. Sounds like you have a lot of other data to keep working on after the expedition. Does anyone else want to add anything? Nope. Okay. So once you got off the vessel and you went home, everyone here spread out throughout Europe and the US. How did you work together to analyze and then write the paper collaboratively once you were off the ship? Does anyone want to take this question?
Maybe I can start it and then Sabine can
Perfect.
continue.
Yeah.
So after all this, after the cruise and then lots of interviews actually, I was super tired. I went on vacation, but during the vacation, I already started to write and get in contact with Sabine. And the first version we wrote together. And yeah, after this, once we were happy with the version we had, we sent it to all the cruise participants and also the pilots of the ROV, and because they were all co-authors on this paper. So that was basically the start, and they gave their input. They all gave their input.
Excellent. Do any of the other co-authors want to chime in about the writing process once you are off the ship?
Yeah, I think Monika already explained how we did it, and also because we already knew the result during the expedition, we had very early on once like an idea of what we want to write down, and that is also the reason why also the paper came out rather quickly compared to when the expedition was, because the story was so clear, and that also of course helps a lot to write the paper. And so basically what is going on right now, so it's basically this is the first paper that explains basically what we found, what we could visually see. And right now, of course, a lot of other work is ongoing because we also collected a lot of samples. So, for example, in my lab, Lara Batista is currently working on the macrofauna and myofauna that we collected also from the subsea floor cavities. And she's comparing now, for example, biodiversity below the seafloor, above the seafloor in much more detail. And so I think in every lab, like people are basically now continuing to work on the samples that we collected. And yeah, there will be, I think, many more papers coming out in the next years to come.
Excellent. Great to hear. Anybody else want to add anything?
Yeah, maybe I can add to this, to all the animals that are identified. And we also have, of course, a lot of samples that were sent off already, and in part they are already sequenced. We're waiting for the last ones so that we have a really overview of all life. So basically, the sequences will tell us what bacteria are in there, archaea, the fungi, which are very important probably also, and then also very tiny little unicellular eukaryotes. So basically they are related to animals, of course, but they're very small and very difficult to identify. So we will have a whole bunch of sequences, including animal sequences also, but also the identification of all the animals. And luckily, actually, we know the site very well. So we will know, once all is basically identified, we will know also, is it all vent animals we knew before? Because or do we find also other species, new species? But this is basically still ongoing.
Excellent. We did have an audience question asking if you found any new to science species in the subsea floor, but it sounds like you're still waiting for that analysis to come back.
Yes, so probably I can give a part of
Yeah.
the answer with regards to the
Yeah.
macrofauna, so animals in the size class from 1 millimeter to like what you can see with your eye. And there are the results what you have so now, but they're not published yet. But what we find in our samples, it's basically that all animals that we see below the seafloor are also known from above the seafloor, from the surface vents. So it really looks that it's that the subsea floor is really a habitat type of the hydrothermal vent ecosystem. And I think that's very exciting that it seems that the subsea floor and the surface vents are very well connected. And it's not only the few species that we already could visually identify during the expedition, but it's actually quite some more that we found. But no new species up to now. But having said, we have no information yet on the myofauna and all the other small organisms. So I would assume, or I don't know, but what we, we got already surprised. So let's see what comes out there.
We'll have to save that for part 2, Back Ashore
Yeah.
part 2. More to come. Very interesting. Thank you. We also have a question on whether you are planning to do some genetic comparisons between these communities and communities at other vent sites, maybe that you have visited in the past or have data from. I think you're muted, Monika.
Direct. So for now, we have no fixed plans for this, and comparisons between different vent communities on the East Pacific rise, or going to the West Pacific, or comparing those communities that are in the Pacific to the Atlantic or some other places. They have been done already, in part, not for entire communities, but for parts of the communities. And of course, this study would be a good background for going to a completely different vent site to compare, because this will be one of the few places, and always has been a place that is very well studied, but this with all the sequencing from individual samples, where we have from one sample everything, that is quite rare actually. So this would be good to compare with something else, but we will see.
Another topic for part 2.
Right.
Does anybody have anything they want to add? You talked a bit about the study site at the East Pacific Rise and how it's pretty active volcanically, but we had a question about what was the size of the study area that you were working within while you were on board Falkor 2.
Yeah, probably I can start. So actually the size was rather small. So we were at the Tinkara vent field, and there's also one area that we call the suburbs. And we actually carried out the experiments in an area like by 20 by 20 meters only. And we flipped like basically 6 times the rocks, and the area that we investigated was like for every sample, every experiment was about 50 by 50 centimeters. So rather small scale. That's also why we're now so interested in also to find out if this is also found, you know, at other vent fields, for example, in other regions, because it's really we just discovered that there is life below the seafloor in these cavities, and I think there's much more to find out.
Definitely. Once the publication came out, you got a bit of attention because it's very exciting scientific news. But could you share with us what the true scientific impact is of this work and this discovery and the publication that came out?
So I think the true impact of the study is that the deep sea hydrothermal vents may not be restricted to the surface, so to the places we can see when we go down with the ROV or with a submersible, but that the subsurface that is hidden can be at least as important as the surface, and that ecosystem hydrothermal vents consists of surface communities and subsurface communities that are connected with each other.
Does anybody have anything to add?
I suppose like another kind of fun thing about it is like it's a slight lesson as a reminder of like perspective in a way. And this is a little bit meta, so excuse me for that. But like, you know, we're surface dwelling beings for the most part, you know, like as humans, and a lot of what we study is like what we can see on the surface. But I think, you know, if you're living at hydrothermal vents where there is no light, regardless, unless an ROV comes down and shines a light, like it doesn't necessarily matter as much whether you're on top or below in many ways, I think. And that's something that we've sort of discovered. And like as we were talking about briefly, like the chemistry and the physical parameters are a lot more important than sort of necessarily exactly where you're seated. And so it's just sort of a reminder that like, you know, just looking at the surface, just looking in the same communities that we might look in the shallow water and other places is going to be restrictive, and we have to think outside the box a little bit. In this case, a little bit, you know, explicitly outside the box, maybe just beneath it, in order to find kind of some really exciting new discoveries and understand a little bit more about life in these crazy environments.
Thanks, Ian. I love the meta. We can certainly get philosophical here. Does anyone want to add anything?
Yeah, I guess, I don't know, maybe even beyond the science. Well, I come from a very small country with little sea coast. And once this work came out and we received a lot of attention, even from our home media, I was really surprised how little people know, even like scientific community, about life on the seafloor of deep sea, you know, and the diversity that is there. And now even like not only on the seafloor, but even below, made us also realize what we don't know yet, and we need to protect for our future now. So how little actually we know about the life on the seafloor. Yeah, I think it was also this part of the importance of our work now is like to open horizon for scientific and general audience, I guess.
Definitely. And my next question was, what do we still need to learn? And we have a question from the audience asking if there was research into the bacterial communities and what do we still need to understand about the bacteria living there. So maybe we can tie those together. What do we still need to learn? And is there anything still that we need to learn or study about the bacteria in this community?
Should I answer? Okay, I start again. Concerning the bacteria and archaea, there are already some publications out there. There are some years old, and one of the co-PIs was on board also. So there is some knowledge already on the subsurface bacteria, but our results, I cannot tell you because they just come out basically of the sequencing machine of the company, and they will be analyzed in very short time, actually, I think. But I don't know if we find. I don't know what we will find. So yes, I mean they are for sure a very important component. So not only the large animals, it's also going down to bacteria. Also the viruses are important, which we studied also. And but the samples are still there. Now we have all the samples, and there's also several smaller projects or so that are still analyzed, because the 20 people that were on board, this was the main project, but there were several other really cool projects that individual people or groups of people on board also worked on.
Excellent. And all of the analysis is still continuing. You mentioned that the paper that came out last October was fairly quick since your expedition was in the summer of 2023, but there's still a lot of work being done, as science is not always quick. And like we say, we highlight a lot of the expedition, but not all of the work that happens afterwards. Does anyone else want to share what we still don't know or what's being worked on still in their lab?
Yeah, probably one part is continuing with the bacteria. So it's, I think, important to also realize that so a lot of the hydrothermal vent fluids exit as diffuse flows, where there's also a lot of bacterial communities associated to it. So, but I think 90% of the heat flux is in the form of diffuse flow at the 9 North area. And also in the cavities, we also found a lot of animals with symbionts with bacteria inside, like all the tube worms. So I think also like the fact that these bacterial communities also occur in high abundance is likely in this subseafloor cavities, also has implications, of course, for biogeochemical cycling.
Excellent. Ian or Tinkara, do you have anything to add?
Sure, yeah. So our sort of one of the side projects on the cruise that I was really focused on was looking at the physiology a lot of these animal microbe symbioses, and really trying to understand sort of how animals and bacteria are able to kind of share these resources and facilitate kind of the survival of both constituents in this kind of pairing. And so we have some really fun, sort of exciting data from incubations that we were able to do on the ship, and some of it we've sort of analyzed. Some of it is still sort of sitting in the freezer. It definitely takes some time, but we do have some sort of exciting kind of new data about sort of the rates of carbon fixation and the rates of kind of oxygen consumption and sulfide consumption of a lot of these symbioses. Some are following up on some of the Riftia work, which is really exciting, and a few of the other kinds of projects are on some really kind of small but interesting little gastropods that are also symbiotic. So it's definitely, there's a lot to learn about both the bacteria and also just how these animals are even kind of working and surviving in these kinds of areas, which is super fun.
Yeah, and if I can just add up that,
Yeah, of
as
course.
mentioned, now not only bacteria or archaea, but also viruses. So we conducted virus production experiments on board, and which are also, as many other things, still being analyzed. But there we try to understand how a viral community there is affecting the dynamics of microbial populations, because, for example, once a virus infects bacterial cell, it can cause lysis. And this is basically resulting in a release of organic material from the cell, which is then a source of organic compounds and inorganic nutrients for the environment. So in this way, also viral populations are affecting the nutrient cycling. And this is also something that, to my understanding, has not been explored before. So it will be very exciting and new data from this type of environment as well.
Awesome. That is very exciting. We have a question from the audience that asks if your findings could be relevant to methane seep carbonate hard grounds, and might there be some assemblages found there? Does anyone want to answer that?
I can try. It's a very interesting question. When we look at the geology, we have different settings with regards to hydrothermal vent ecosystems and seep ecosystems. So, for example, in 9° North area, it's along a mid-ocean ridge, where we have hard substrates. In these areas where we worked, there were also like no sediments. We also have this, volcanic eruptions that spit out lava, fall back again. During this process, you have evaporation and this formation of these cavities. Whilst when we look at the seep ecosystems, they are geologically older. They're typically covered with sediments. What you do get over time is that you get carbonates. So I don't know if there could be something similar related in there. But I think like what we found now as in the subsea floor cavities at vents, I would not expect that we find this at the seep ecosystems. However, what we also need to acknowledge here that when we look at seeps, for example, there are also like tube bombs there, lamellibrachia, that also have roots that also reach into the sediments, and also we have life in the sediments at seeps. But I would not expect any subsea floor cavities at seeps. But I don't know what Monika or others here think about that.
Also, similar cavities I would not expect, because those carbonates, they develop as clumps basically that can grow, but I don't know if there can be cavities inside these clumps. And even if there are cavities inside, you would need to have an exit, also praktisch an entrance to it. So in this respect, I think they're very different. So if I would go to the seeps, I would look at what lives in the sediments and how deep basically life into the sediments occurs, because as long as animals need oxygen, so you go into the sediment and maybe you reach a carbonate or not, but the limit here would be that for animals you need oxygen, and after this it's anoxic, and you can find then basically microbes you can find everywhere. And it is known that you can make, there are many sediment cores taken, and the microbial community is very well studied.
Excellent, thank you. That wraps up our audience questions. If there are any other audience questions, feel free to get them in. We have about 10 minutes left, but while we wait to see if any audience questions remain in our last 10 minutes, I know you each had already talked a little bit about waiting for some of the genetic analysis to come back from the lab and some other papers that you have in the works. But I just wanted to give you another chance to talk about what you're working on now from your expedition. If you still have questions that you are trying to answer from the samples that you collected, or anything else that's kind of happening in your scientific life post-cruise from this expedition. Tinkara, do you want to start?
Oh. I'm probably most far away from the field. Yeah, well, I participated to the cruise after Monika invited me. It's not like really my primarily habitat to investigate. So it's a very kind of new thing to me, but the analysis actually has already been conducted in terms of a wet lab. So basically really like analyzing the samples on the machine. So now it's about the post-processing or like statistical analysis and so on. And in the process of writing, in fact, by my co-author, together with my co-author from University of Vienna as well, Christian Winter. So I do see Monika a lot because she comes to our institute quite often, visits Slovenia, and we get a chance to discuss the data and the paper, and well, hope to visit you soon again in a month or so. When we can hopefully have already a draft of manuscript ready to read. Yeah, so that's from my side currently from that expedition. Of course, as a scientist, you always have 10,000 things popping up. So
Excellent. Good to hear. I hope you get to see Monika soon and work on that paper. That it's always nice to be able to do those things in person, since we've become very reliant on Zoom, as we discussed. Ian, is there anything you would like to share about some of the data that you're working on?
Sure, yeah. So a lot of the kind of side work that I was doing on this this cruise was focused on a very sort of slightly strange group of gastropods, so snails and limpets. And many of them are pretty poorly categorized. There's maybe like one or two papers ever really written about them. And admittedly, unless you love gastropods and limpets, they're very small, and you know, they sort of, some of them are kind of nondescript. So I think they haven't gotten a lot of love. But for that reason, they're sort of right for the you know, asking questions about how they evolved and what they're doing. And so I was able to collect quite a few different species and genera that that have most of them have been described. I think all of them I've gone through. But there's little to no genetic data available for several species and genera that I was able to sample. So I'm kind of combining some of the samples that I got from that cruise with museum samples and some existing data that's already published online to try to put together a pretty robust kind of phylogeny. So an evolutionary tree of this group of gastropods called Neomphalionis that are pretty much endemic. Well, this particular subset are really endemic to hydrothermal vents. So it'll be exciting to see. I'm kind of still amassing data from collaborators in different vents around the world. So it's been really kind of fun. But I'm definitely one of the main takeaways from that sort of side project on this cruise. We'll be having a bunch of data that I can put together and a bunch of new species I can add to this this larger data set.
Awesome. Please keep us updated on all of that work. And I'm glad someone can show the gastropods some love, as you put it. Sabine, would you like to share what you've been working on?
Yeah, so Lara, who's working in my lab, she's right now analyzing the macrofauna and the myofauna. And actually, next week we're going to Hong Kong to Deep Sea Biology Symposium, where Lara will present a poster on the findings. Next to that, from a student of mine, Alessandro, who was also on board of the vessel, he worked with high pressure vessels and did limits of life experiments, what kind of temperatures small copepods can tolerate. Also there we have some really good results. Also his master thesis is finished, and now it's basically also like the finishing touch for the paper. And so this is ongoing work. And I think regarding the future, I think we are all thinking about what's next. So yeah, you know, how can we explore where this, so if this vent subsurface habitat is present like globally and to what extent. But it's more like a, you know, current, yeah, thinking about what methods we could use in the future to make some more groundbreaking discoveries, I guess. Yeah.
Excellent. Well, I hope you have a great conference next week with presenting the work with your student. That's very exciting. And Monika, I think we'll save the last word for you. Would you like to talk about what you are still working on from this expedition?
So the only thing I will personally do is I will go through the bacterial data set once they are sequenced and check out if the symbiont of the giant tubworm is in there, which I expect, but this I would like to confirm. And other than that, I wait for all the data, because then we will bring a lot of people back together for the next big paper on the entire community and the connectivity between subsurface, surface, and the surrounding. So this will be also a very large paper with a lot of data, but it will take still a little while. And other than that, I would just like to dig a little bit deeper and more, actually, what Sabine said. Now we're thinking about it because the subsurface has layers of these cavities. Basically, you see a cavity here now, but there's another cavity below, and another cavity below, and another cavity below. And of course, it will go warmer and warmer, and probably less and less oxygen. But we don't know that. There could be another layer of animals. And so what I mean with this is it would be worth to develop tools to go to such places and to dig deeper and check it out. And also, of course, to look at the horizontal extension. How far do they extend those subsurface hydrothermal vents? Yes.
Thanks, Monika. Please keep us updated on that analysis. And when the next big paper comes out, maybe we'll do a Back Ashore part 2, since we alluded to a lot of more exciting results. And I love your additional questions. It sounds like that might be another proposal that you also have to write to get some ship time. So I wish everyone the best of luck in all of their analysis. And I really want to thank you for taking the time to speak with me and our online audience about what you have been up to since you stepped off the vessel in 2023, and congratulate you on your paper and your groundbreaking outcomes that really received a lot of attention and were a big deal in the scientific world. So thank you again. Happy New Year. Thank you, everyone online, and we will see you soon.
Thanks a lot.
Thank you. Bye.
Thank you.